Welcome back, Greenhouse Science. Today we're going to talk about cell biology. We're going to talk about what kind of cells we're looking at in animal and plant cells and how these cells are made, the stuff inside of them. Very exciting topic. So all living organisms have cells. Cells, the basic unit of life, smallest unit that can carry out life's processes, and they weren't discovered until the 1660s. So we didn't know about them for most of human history. But cells are the most foundational part of what makes an organism alive. Robert Hooke, he's credited with finding the first cells. He was actually finding the cell walls of plants, which is very fun for our plant science class here. But he was studying nature using a light microscope. He looked at a very thin piece of cork, and he saw that top image here, where he saw a ton of little boxes, and they reminded him of rooms where monks lived. And these rooms right up here above me, that is what monk cells looks like, and that's what cells are named after. Anton von Leeuwenhoek was the first person to observe living cells. His lenses were about 10 times more accurate than Hooke's were. And in 1673, he observed cells with these green stripes in them called spirogyra. These guys right here above me. He called these guys animalcules, but later we called them protists. They're kind of a junk drawer of organisms that don't fit into plants and don't fit into animals, kind of fit into a middle ground there. We started to have a whole bunch of people really excited about cells, talking about cells. We came up with some things, some ideas. Matthias Schleiden says, hey, we have a plant, it makes more plant cells, so all plant cells came from plants. Animals, Theodor Schwann says, hey, all animals are made of cells. Like at the basic smallest level, they have cells in them that make them work. And then in 1855, Rudolf Virchow, he comes along and he says, hey, all of these cells are coming from pre-existing cells. So all cells must come from other cells. Bacteria, animal, plant, just spontaneously happen. All of that went into the cell theory, and this is the cell theory here. You have all living organisms are composed of one or more cells, that the cells are the basic unit of structure and function in an organism, and that cells only come from the reproduction of other cells. You can't get just cells, like we said, coming out of nowhere. Not all cells look the same. There's a lot of diversity amongst them, and if you have a differently shaped cell, it probably has a different function. So in human cells, we have some that protect the body, send electrical signals. Our blood cells carry oxygen. In plant cells, you have ones that provide structure, that move water up and down the plant, or ones that like guard the stomata and open it and close it so that that plant can breathe, basically, in carbon dioxide and out oxygen to do photosynthesis. Not all cells are the same size. You have really big cells like ostrich eggs, chicken eggs, giraffe nerve cells are like 6 feet long. But then you get really small, like a human egg cell, the size that we were when we started out, is the size of a period. And even smaller than that, you have your normal, typical plant cells and animal cells. Most of the cells that we see are microscopic. I mean, we can't see them with the naked eye. We have to use a microscope or some other aid to get down to that level to see them. Cells have to have a certain amount of surface area to volume ratio. Surface area is the amount of stuff that touches the outside. They have to be able to touch the outside to get nutrients, to get energy, and they have amount of stuff inside of the cell, the volume. The ratio of this has to be nice for the cell. It has to be a certain ratio so that we can get things in, and then it doesn't get stuck inside of there because there's so much volume on the inside. It can find its way back out too. So the surface area to volume ratio is really important. Some things that we need to get inside of the cell are things like oxygen and nutrients. Things that we need to get out are things like waste. And so we will move them in and to and out of the cell there. In a very large cell, your volume would be too much to get all the nutrients you need, and it'd be too much to get rid of all the waste. So you can't have too big of a cell. Cells kind of have to be start out a certain size. All cells have three main regions that we kind of break them into. We have the nucleus, the cytoplasm, and the plasma membrane. You can get into those a lot more, but those are kind of the three basic parts. The plasma membrane is a fragile, transparent barrier. It keeps all the things inside of the cell, and it kind of creates the skin of the cell that separates the outside of the cell to the inside of the cell. So make sure that things that are supposed to be on the outside of the cell stay on the outside, and things that are supposed to be on the inside stay on the inside. We also call it the cell membrane. It is a dynamic changing thing. It is not a static, boring thing. It's got two lipid layers uh, that kind of make up a bilayer there. And we also have cholesterol in high concentrations in the plasma membrane. And where normally you hear cholesterol, you think cholesterol is bad. Cholesterol helps to keep membranes fluid and flexible so they don't break when they're sliding and slipping past each other. The cytoplasm is a region of the cell that includes everything except for the nucleus. So it has organelles, fluid, and cytoskeleton in here. It's not just liquid, and it's more like a gel. Um, of that, the fluid part is called the cytosol, and it has lots and lots, thousands of dissolved molecules and particles inside of it that the cell is able to use for energy and cellular processes that it needs to. The nucleus, not all cells have a nucleus. Some of them, like bacteria, have their DNA free floating, but all plants should have nucleuses. And it's the headquarters of a cell. We also call it the control center. It holds and contains all the DNA that a cell uses. This deoxyribonucleic acid contains all building instructions for the body of a plant, the body of a human, the body of a dog, the body of a cat, the body of a fish, etc. When we talk about cells, there's two types of kind of categories that we break cells into. The first is prokaryotes. And these don't have a nucleus and they don't have organelles. They contain their genetic information in a part of the cell called the nucleoid, meaning it's nucleus-like, but it's not a nucleus. We further break the prokaryotes down into two groups, the bacteria and the archaea. We'll talk about those briefly. Bacteria is all organisms that are similar to the first cellular organisms, and archaea is organisms that are more closely related to modern eukaryotic cells. So broke off a little bit after bacteria. The second type of cells are eukaryotic cells, and these are organisms that can be made up of one or more cells. They have a nucleus, and they have membrane-bound organelles, things like chloroplasts and mitochondria, nucleuses, endoplasmic reticulum, that kind of stuff. Organelles, these are structures that are smaller than a cell. They are usually surrounded, some of them are surrounded by a membrane, and they help to carry out cellular processes. Because of these things, eukaryotic cells can get and typically are larger than prokaryotic cells and what we would call more complex. Cellular organization, we can have some colonies forming. This is a collection of genetically identical cells that live together in a connected group. It's not a truly multicellular thing because their cellular activities are not coordinated as an organism. They're still very selfish, out for just us. Once you get to true multicellularity, your cells become more specialized. And then eventually you have to be in a group and you can't survive independently. So when we have groups of cells carrying out a specific function, they're called a tissue. Tissues make up organs, organs make up organ systems, and organ systems all together make up an organism. Many organelles have a phospholipid bilayer. The hydrophilic end is attached to water, and the hydrophobic end doesn't mix with water. It only touches another hydrophobic tail there. This, however, makes this nice bilayer or double layer of phospholipids, and that we call the fluid mosaic model because the cell is both strong and able to slide, and it has a ton of things inside of it, things like glycoproteins, carbohydrates that you can see on this picture up here, that it looks like it has a lot going on. It's not just a static, boring thing. Membrane proteins, these can be embedded inside of the plasma membrane. They can send and receive signals. Your blood type is a membrane protein. Plants have membrane proteins. They also help transport materials into and out of the cells. They have these pores and carriers that help move things from one place to another. The nucleus, it can be broken down into three main parts. It's got the nuclear membrane, nucleoli, and then chromatin. The nuclear membrane is a double-bound membrane. We call it the nuclear membrane because it surrounds the nucleus. It's a lot like the cell membrane, but it's a nuclear membrane. Between the two membranes, there's some fluid, and then on either side, we have some fusion that forms. You can see like these holes here. Get my pen out. Okay, here, these are nuclear pores that can form. And this helps to let things go into and out of the nucleus versus the cell, where it's much more tightly regulated what can go into and out. Like we said, this nuclear membrane is selectively permeable, and it's much more freely permeable because it needs to get the information from DNA out into the cell to make protein, to make whatever is going on for that cell. Inside of our nuclear membrane is a jelly-like substance called nucleoplasm. It's a lot like cytoplasm, except it's inside of the nucleus, so we call it nucleoplasm. Nucleoli, these are small, dark brown bodies inside the nucleus. They are where we make ribosomes for the cell. And ribosomes are the sites for protein synthesis in cells. And protein synthesis is a big reason for how plants are able to do what they do. The next thing is chromatin. When a cell is not dividing, its DNA combines with protein. This forms a bumpy network called chromatin, is the DNA plus this protein. When the cell is dividing, this chromatin condenses to form rod-like bodies called chromosomes. And you can see some chromosome up here. But makes these chromosomes that we can then see. But most of the time, DNA is in this loose, kind of loosely coiled form. Ribosomes, these are little tiny, dark, round bodies inside of the cell. They make they're made of proteins and RNA. The RNA that they're made of is called ribosomal RNA, named after it because it makes it. This is where protein synthesis actually happens. And some of these are floating around in the cytoplasm, and most are attached to what we call the rough endoplasmic reticulum. Endoplasmic reticulum is a system of fluid-filled canals and cisterns and tubules that coils and twists through the cytoplasm, and about half of the cell's membranes are inside of this structure. But what this does is it provides a network of channels for us to carry substances through, and the primary stuff that we're carrying through here is proteins. And as it goes through here, we finish it off, make the protein good, put proteins together to form molecules, then we'll go into a cell to do a job. The rough ER is the endoplasmic reticulum that has lots of ribosomes on it. It has all of the cellular membranes forming inside of the rough ER. It's the membrane factory for the cell. On these ribosomes, we make these proteins. They then get folded into a 3D structure and sent off into the cell to do whatever job we need it to do. The amount of rough endoplasmic reticulum you have is an indicator for how much protein you produce as a cell. So it's cells that produce a lot of protein, like the pancreas in humans, have a lot of rough endoplasmic reticulum. Those that don't make a lot of protein have less endoplasmic reticulum. The smooth ER, this is on the outside of the rough ER. It's a continuation of it. It doesn't do any protein synthesis, but it does do some other things. It makes cholesterol, it metabolizes fats, and it detoxifies drugs. In humans, we have a lot of smooth ER in the liver because that's where a lot of this job is being done. Golgi apparatus, this is a stack of membranes that make these little vesicles, which are tiny liquid capsules. These guys are what we store protein in, and when we're shipping it across to other cells, to different parts of the cell, usually the Golgi apparatus is close to the nucleus, close to that rough endoplasmic reticulum. It's going to modify and package the proteins and then decide where they need to go inside the cell. These vesicles that we make, secretory vesicles, we put synthesized proteins in here. They can travel to the plasma membrane, and then they can fuse with that membrane and inject the material out of the cell. And plants also have hormones that they use, and they get ejected in the same way. In addition to secretory vesicles, the Golgi apparatus makes lysosomes. Lysosomes or breakdown bodies is what that term means. But their job is to digest things inside of a cell. They have bags inside of them that have digestive enzymes. They function as a demolition site for cells, for bacteria, for other things. In humans, they're really abundant in white blood cells. But they also can rupture and digest the cell if there's not a lot of oxygen present in a cell. So some other functions there. Peroxisomes, these are only found in animal cells, but these are membranous sacs containing a powerful oxidase enzyme. They use molecular oxygen to detoxify harmful poison substances like alcohol and formaldehyde. They also disarm free radicals. These are highly reactive chemicals with unpaired electrons that have the potential to scramble protein and nucleic acids. So getting these guys neutralized is really important. Mitochondria, all plants have mitochondria. They're the powerhouse of the cell. They have a double membrane where the outside is a smooth membrane that offers protection and like kind of lets it slide around inside the cell. And then the inside has these shelf-like things called cristae. And this cristae here are these shelf-like projections that help increase surface area so that we can do more energy production inside of mitochondria. There's enzymes inside of the mitochondria that carry out reactions with oxygen. We break down food with them to produce ATP molecules. ATP, or adenosine triphosphate, produces all energy for cellular work. In humans, liver and muscle cells have a high number of mitochondria, and stuff like fat that's inactive does not have a lot of mitochondria. Cytoskeleton, this is a network of protein structures in the cytoplasm. It helps to support the cell's shape, it helps to support organelles, and it provides machinery to move things around inside of the cell. From largest to smallest, we have the microtubules, intermediate filaments, and microfilaments inside of the cytoskeleton here. Microfilaments, they're actin and myosin. They help with cell motility. They also provide cell shape change. They do it by walking on this track here. This is a very cool diagram that shows that happening there. Very fun. Microtubules, these help determine the shape of the cell, help distribute organelles, and are very important for cell division. The centrioles, these lie much closer to the nucleus. We find them in pairs. They're often rod-shaped bodies that are right angles to each other, and they're made up of microtubules that help split the cell later. These are not in plant cells, they're more common in animal cells. Same thing here, cilia. These are whip-like extensions that help move things along the cell surface. They're found in the respiratory system to help clean airways, and they're formed from centrioles. They're present in animal cells, but not in plant cells. Same thing with the flagella. Well, flagella do exist in some gametes of plant cells. When we get long projections of centrioles, they're called flagella. In human body, we only have sperm cells that have a flagella. The flagella helps move the cell through an environment. Cilia helps move things across the surface of the cell, not so much helping the cell move through the environment. Plant cells have most of the same organelles. They have 3 additional. They have cell walls, large central vacuoles, and they have plastids. Things they don't have are the lysosome, centrioles, peroxisome, cilia, and flagella. The cell wall is a rigid layer outside of the cell plasma membrane. It has a polysaccharide called cellulose that is not made in animals, but it forms a box around each cell, and that's actually what Robert Hooke was seeing when he was looking at that through that microscope. Cell walls also have pores in them. This helps allow water, ions, and molecules to enter and exit the cell and makes it less of an impermeable barrier there. There's 2 types of cell walls. There's primary and secondary. Primary cell walls are formed by enzymes that are directly on the plasma membrane, and they grow in one direction out. Secondary, when the cell stops growing, we get between the plasma membrane and a secondary cell wall. It is very strong, but once it forms, it cannot expand anyway. So secondary wall will form when the plant is adult plant. So central vacuole, this is a large fluid-filled organelle that stores water. In this one here, it's right here. This is your central vacuole. But it stores water, and inside of that water is dissolved enzymes, metabolic wastes, and other materials inside of there. Usually they're formed as we have small vacuoles coming together, fusing to form a larger one, and it can be up to 90% of a cell's volume, and it can push all those other organelles to the sides there. Like you can kind of see in this picture in the top there, up there, there's lots of organelles being pushed away from the middle because of this central vacuole. Water has a profound effect on plants and their cells. When water fills a vacuole, the cells expand and the plant stands upright. When it loses water, the cells shrink and the plants wilt. So we try to keep our plant cells full of water. There's other vacuoles that exist in the cell. They can be used to store toxic materials. Acacia trees are a great example. They store poisons that protect them from predators. And then vacuoles can also store color pigments, like in carrots. They have carotenoids they have a lot of carotenoids in them that are stored inside of vacuoles. There's your acacia there, your Minecraft acacia. Plastids, these are organelles like mitochondria, but they're a little bit different. They're more like chloroplasts there. They have a double membrane. They contain their own DNA. There's lots of different types. There's leucoplasts and protoplasts and chromoplasts, amylioplasts, and then chloroplasts is the one you're probably most common with, but it is an example of a plastid. Chloroplast uses light energy to make carbohydrates from carbon dioxide and water using thylakoids, which are flattened membranous sacs. They have a green pigment in them called chlorophyll, and chlorophyll captures and absorbs light. And these guys can be found in both plants and eukaryotic algae like seaweed. That's not a plant, but probably a pretty close relative. Chloroplast DNA is very close to that of photosynthetic bacteria, and chloroplasts only come from other chloroplasts. Because of that, the evidence suggests that chloroplasts are actually descendants of photosynthetic bacteria prokaryotic cells. How we got them inside of cells is through a process called endosymbiosis, where ancient prokaryotic cells were incorporated into plant cells. These cells then entered into a symbiotic relationship. Chromoplasts are another example here. They contain colorful pigments that can or cannot be used for photosynthesis, like this carrot up here. That root that's orange is not getting a lot of photosynthesis happening. But it is a carotene, an orange pigment that gives carrot their color. So while it doesn't require photosynthesis, it is something that the plant uses the vacuole for. Last thought here is there's some other plastids. They generally have the same characteristics as chloroplasts, but they differ in what's inside of them. Things like amyloplasts store starch, and all of these plastids are coming from a common precursor called a protoplastid. And think like a stem cell, but for these plastids. So they're all kind of forming the same way, and then they differentiate as they get older. That is all we're going to cover in this one. Thank you for watching. I hope you learned something, and we'll see you in the next.